Okay, so you guys should be or have been learning about the organelles, right? Kind of on your own time, and hopefully you're starting to, understand or know and learn what each one of the organelles do. But what our focus is going to be today is two things. How do the organelles work together, right? So a lot of times we learn the organelles in isolation. Oh, the Raphael makes proteins. Oh, the Golgi body does this. What we want to try to do is figure out how these things are sort of working together in systems, okay? And then the other thing is how organelles really help to lead to different types of cells, right? So how do those subcellular components, by the way, when we talk about subcellular components, what are we really trying to say there? It's a fancy way of saying what?
More than one cell.
Not more than one cell. Think the opposite. Don't go bigger than a cell. Sub means below or beneath. So we're talking about what's lower than a cell. So the organelles, right? Just like if you were to talk about subatomic particles, we're talking about the proton, neutron, electron that make up the atom, right? So cell types and subcellular components. So how do the organelles help lead us to different types of cells, okay? So two things, how they're working together and how the organelles lead to different types of cell types. What we're going to do is we're going to look at some sample questions from the College Board, okay? So let's start with that. Okay. So the first question here is going to deal with what we call the endomembrane system and focus on how do organelles work together. I picked this question to do with you guys in class because when I assigned it last year, I noticed a lot of students had struggle with it, and I can understand why, because we now have to make that leap from how do the individual organelles, how are they all tied together, right? So let's look at the question first. The figure above shows a series of microscopic images taken over time. You can see we're progressing in time here. Of ovalbumin production in chick oviduct cells. Okay, so we're looking at a specialized cell here. And it's in response to stimulation with estrogen. The ovalbumin protein was detected using a fluorescent dye as shown by the white areas in the time-lapse sequence. So anywhere that I see white up here, that's where I see the protein, okay? So take a look at the images over time and notice the white. So in this first image, do you see any white?
No.
Where does it start to show up here?
Around the middle.
Around the middle, okay? So you start to see it sort of in this network outside the middle. And that's one cell right there, right? So you can kind of see one full cell and then a piece of two. All right, how is this one and this one different? What do we notice here?
It's going up.
Okay, I start to see it expand this way. And in this one, I can really see like circles of it, right? The white. And then eventually it's going out more, okay? So that's what we're looking at. Now, what we're gonna do is we're gonna finish this sentence, okay? So the microscope images indicate that the ovalbumin, which again is a protein, okay? We wanna know, okay, the microscope image indicates that ovalbumin, you gotta finish the sentence. What I want you to do is just take a minute and a half, read through the different options and pick one, and then we'll share to see which ones you guys came up with. in the picture? We're back here.
So in like the last picture where they're like all connected at the top, is that the membrane?
Nope.
Nope.
That's going to be, no, that's going to be what we call an extracellular environment. This is sort of outside of the cells.
So the membrane is like the lining between of them, right?
So when you say membrane, what membrane are you talking about? There's a lot of membranes going on here.
The cell membrane.
The cell membrane is right here.
Oh, okay.
So this original cell, like I said, this is one cell right here. And then you can kind of see pieces of it, the next one. And then this is outside of the cells. The extracellular environment. Good question. Trying to figure out what you're looking at. So, do you guys come up with a guess yet?
I kind of just looked at the words and kind of assumed. I kind of just guessed it.
Okay, so what, if that's fine if you're just guessing for now. That's why I'm going over this particular one with you guys. So which one did you guess?
See?
You're guessing C.
B.
B? B? Okay, so we got 2 for B, 1 for C. All right, so let's talk about some of the other ones. Why did you not pick A? Like what? So let's dissect it a little bit, okay? So the ovalbumin, which we know is a protein, okay, is synthesized in the ribosomes. Does that make sense? Do ribosomes make proteins? Okay, so that part of it's correct. And then it says is packaged into lysosomes, which is then secreted into the oviduct, which is that extracellular space we're talking about. So what do you guys think about this phrase, packaged into lysosomes? Is that a correct statement?
No, you can't package it.
Well, you can, but is it going to get packaged into lysosomes? Why not? What's the job of a lysosome? This is where you really got to know your organelles too, right? So a lysosome, what's the root word there?
Like,
Which means to, otherwise known as lysis. We've seen that before. What did we say lysis means to do?
Separate.
To break apart, separate, break down. Why is a lysosome called a lysosome? Because what does it do? It breaks down things, right? It gets rid of waste and breaks things down. I know in my AP assignment that you guys had, there was a couple of questions with lysosomes involved. And you may have heard the phrase hydrolytic enzymes. Hopefully you guys remember hydrolysis is when we were using water to break apart the polymers into their monomers, right? It was the opposite of the dehydration synthesis. So there's that root word lyse again, meaning to break apart, and hydrolytic enzymes. It's got to help break things apart, okay? That's just a refresher on lysosomes, but we know that this is now not the correct response because lysosomes really have nothing to do with protein production, okay? So that's why A is no good. When it comes to D, I'm going to actually come back to D because it'll make more sense once we figure out, you know, whether it's going to be B or C, okay? So in B, we're saying it is synthesized on the ribosomes that are bound to the rough ER. That's a true statement, right? We can make proteins on the rough ER because that's where ribosomes are. And then it says that it's secreted directly into the oviduct, okay? So we're saying this is secreted directly into the oviduct. So that's one option. In C, we're talking about the protein, crosses the endoplasmic reticulum as it's translated. That just means that as it's made, it's leaving the ER. That's an okay statement, right? It's a different way of saying this. Then it moves to the Golgi body, okay? And then to vesicles from which it is secreted. So now, think about what is the purpose of the Golgi body? What does the Golgi body do? So can I just break it down a little bit more, right? In this one, we're going directly from the protein being made at the R to outside of the cell. But in this case, we're having the protein being made. It has to make a stop at the Golgi body and then vesicles to the outside of the cell. So what is it that the Golgi body does? Does that make sense? Does that idea of this protein's got to stop at the Golgi body make sense?
So it's like a pit stop?
It's a pit stop. Why? You guys got to tell me what happens at the Golgi body. Got to know your organelles. Somebody look it up. What's the Golgi body do?
Packages proteins into vesicles.
Aha. So it packages proteins. It modifies them. In one of the videos that you saw, it should have talked about how one of the ed puzzles that it modifies it so that it can be used by the cells, right? So does it make sense that a protein after it's made stops at the Golgi body to be modified, and then what? It has to be packaged into vesicles for secretion. So C is our correct answer. Okay? And we're going to go back and look at the image to show that, okay? But let's talk about D for a second. If our protein is made and it crosses the Golgi body post-translationally or after it's made, and then it's taken up by the Golgi apparatus, what it's saying is it's going to, the protein's made in the Golgi body, it goes to the Golgi body and endoplasmic reticulum, and then is packaged into the secretory vesicles. I don't even know how you say that, but this one doesn't make sense because it's kind of got the Golgi body and the endoplasmic job reversed, right? It's almost starting with the Golgi body and then going to the ER, and that's not how it works. It starts with the ER, then the Golgi body, then packaged for export. So let's go back and look at the picture and see if that makes sense. Okay, and we'll identify some of the structures here. So our initial cell, I'm trying to find a marker that works here, but no protein going on, right? So what is this network right here? What do you think that is when the protein first starts to show up? That's going to be your ER. What's the circle in the middle then? What do you think that is? So if the ER is making the proteins, who gives you the instructions for what proteins are made?
The nucleus.
So we got our nucleus. By the way, endoplasmic reticulum is always found right outside the nucleus, because this is the instructions, this is the manufacturing, okay? Now where does it go? We said, oh, it kind of went up a little bit, right? So we have this network of the ER. What do you think this is right here? Nope, that's this.
No. Oh, okay.
It goes from the rough ER to the, we just said it, the Golgi body.
Yeah.
All right, so here we got our Golgi. This marker stinks too. And if you remember what Golgi body looks like in some of the images you saw, it kind of has that like look to it. You can kind of see that there. Okay. From the Golgi body, what are these round circles? Vesicles. And then from the vesicles, what happens to the protein? It gets released. It's transported out of the cell, right? So now we're back to our external environment. Or what they are calling the oviduct. Okay? So again, this is where we're trying to point out that these organelles, they don't work in isolation. They're part of a bigger system that's going on, right? And you have to be able to connect one to the other. It actually will help you in understanding what the job is of each one instead of trying to remember what does that one do. If you think of it big picture wise, right, and how proteins are made and shipped out, then you know each step along the way that it's got to take. So it's almost better to think of it in terms of a system and the overall big picture than isolated individual roles. You know what I mean? All right, so that's the first one. I'm talking about how organelles work together. Then we want to tie in this idea of subcellular structures and how they lead to specialized cell types. So I know you guys don't have this one in your PowerPoint. I added this in after the fact as a sort of a segue into the next couple of questions. But here's an image of different types of cells in the body. What do you, without even reading this, what do you notice about that picture?
They all look different.
They all look different. And from what you know about them, not only do they all look different, but in terms of their function, they all have different functions, right? So, oh, wait a minute, where have we seen that before? Structure, function, right? The structure might tie into their function. So there are actually 220 different types of cells in the human body. Each cell type has a unique structure that is related to its specialized function. Okay, so now we're just looking at overall structure right now. And so let's examine specialized cell structures. The paragraph here tells you that in multicellular organisms, cells are specialized to perform various roles within an organism. Therefore, a particular kind of cell may contain large amounts of certain cellular structures or large amounts of organelles that allow the cell type to perform its specialized role. Meanwhile, this kind of cell may actually completely lack other organelles that are not needed for its role. Okay? So you're going to examine the cells below, and we're going to use the table to sort of record how their structure might tie into their function. What happens is when you guys are taught the cell, middle school, freshman year, you're always given this perfect image, you know, of perfectly placed organelles, and an animal cell is always this round thing, and a plant cell is always this box-like thing. Where in reality, do cells really look like that generic cell that you're used to taught with? No, right? Not at all. So we want to now think, okay, how are these cells different from each other that allow them to do their function? So just making observations, what do we know about the structure? We've got muscle cells, nerve cells, and blood cells. How are they different?
The blood cells are like circles.
So our blood cells have this nice round disc-like shape to them.
The nerve cells are look like spiders, kind of say.
So like a web, almost like a network, I'm gonna say. They have like the cell body, but they have these long things that kind of reach out and touch one another. And then both of those are very different from what our muscle cells look like, right? So how could we describe what the muscle cells are?
Stretched.
They're like elongated, flattened, and notice how tightly packed they are, right? So let's use those observations and talk about how that might tie into their function. So you already started talking about this in terms of what do muscles do? They stretch, they contract and stretch, right? So do they need to be able to elongate? Yes, right? So that helps with the muscle contractions. And the fact that there's so many of them packed together
Means it's solid.
means it's what?
Solid.
Solid, and maybe packs more of a punch, right? Does more work that way. Think about what muscles do. They have to lift, they have to exert, they have to do a lot of work, right? A lot of power. So the more of them I have in a certain area, the more powerful they're going to be. So we've got that elongation that allows them to stretch, but we also have that density that allows them to be more powerful. If you guys want, we're kind of working our way through this. I just have my screen up. So we've already talked about their differences in appearance. Okay, so we know that the muscle cell, elongated but densely packed, is allowing them to contract and stretch and give more power. Okay, how about the nerve cells? How might the structure of these nerve cells relate to its function? What do we know nerve cells do?
They control their movements and memory.
They control movements. Memory is a big part of the brain, right? So if they're controlling movements, your brain has to somehow be able to tell, me to move my hands, right? Or that's the one that I know, but your brain is also telling your heart to pump, right? So it's constantly sending out messages that you either have control over or you don't have control over, right? Or if you feel something, there's nerves that pick up that, sensory neurons that pick up that message and send it to your brain. So you're saying, oh, this is, a hard surface. Oh, my hand's near heat, right? So it's constantly sending nerve impulses or messages back and forth. So how does that structure that we're seeing help? Would that help to send messages?
Yeah, because since they're connected, it like sends them like through the connected.
Right, and it's kind of the way that thin piece reaches out and sort of contacts the other neuron makes it really efficient, right? Because imagine if you had to send a message through something that was really big or thick, right? It's got a lot of space that it has to travel with. By making those like thin connections, it kind of targets where it's going, and makes it go really fast. The fact that they're all connected that way too. Okay, so it helps you send nerve impulses by allowing messages to go quickly and throughout sort of that network that we were talking about throughout your body. I think you mentioned the word web, right? And I think of that almost like the World Wide Web that connects, can send messages like crazy. Signals and things back and forth around the world. They don't refer to the internet as the web anymore. I don't hear it that. Do you guys ever hear the internet referred to as the web? Did I just date myself with that? We used to call it the web. You know what WWW stands for? The World Wide Web. Okay, phew. All right, at least you guys knew that one. You didn't know that?
I know what AOL means.
Oh, what's AOL?
America Online.
America Online. Okay. Yeah, the web. Oh my gosh.
You just say the internet.
The internet.
The intro.
Or the Wi-Fi.
But it makes sense, right? The web, constant messaging going back and forth. All right, let's do the last one. How does the shape of the blood cell help do its function? What do you think?
It's like loose, not loose, but it's not compact, so it allows for the blood to be liquid. The fact that they're far apart, where the muscles are close together.
Okay, so the fact that they're not really connected to each other, right, and that they're a little bit further apart allows that blood flow, is what you're talking about. And what do red blood cells do besides flow throughout the body, or what's the purpose of having them flow?
They collect nutrients from areas and transport to other areas.
So what's one of the biggest nutrients that they carry for us?
No.
Your red blood cells carry oxygen. Good job.
Look at me.
Right, so they carry oxygen, and so notice the little disc shape there. The oxygen actually sits right in the center there. It's like a little holding thing for it.
Like you sit there.
Yeah, it catches a ride, right? It's almost like a floaty. Like in the pool. That's all I can think of right now is the oxygen sitting in floating down the bloodstream. Okay? But there's a reason why it's in the shape of a circle too. Think about that. When you go have you ever been down the lazy rivers at the water parks or whatever? They give you a flotation device that's in the shape of a circle, not a square. Right? Why is it in the shape of a circle?
It spins, it rotates.
It spins, it rotates, the flow is easier. Imagine if everyone had these sharp edges that were pounding into each other, you'd get bumping into each other and getting stuck and clogged. So that circular shape helps with the flow. Things can sort of slide by each other a lot easier.
Wait, so how do blood clots work? Because that's when there's too much blood, right, in one area?
Yeah, so blood clots can happen for a lot of reasons. It could be, there's one particular disease came up in the last class. Have you ever heard of sickle cell anemia?
It's when the roundness of the cells get flat.
They instead of having this nice round structure, it becomes more of the edges of it get kind of pointy and it's flattened, and it looks like what they call a sickle, or if you've ever seen like the Grim Reaper, and he's got that thing, that's what they look like. So it's got these like sharper edges to it, and it prevents flow, and so the things kind of get caught with each other on different things inside your body, or inside your veins and things. So that's sickle cell, so that can cause blood clots. Other things like buildup of cholesterol and things in your arteries can cause blood clots. The weakening of blood vessels, like brain aneurysms, is basically when you have weakening of the blood vessels, and it creates like this bulge in your vessel, and so the blood kind of gets stuck inside there, and eventually could pop. That's what lead to, that could kill you too. So that could lead to a blood clot. So there's different reasons for blood clots, but ideally we want this nice flow with these nice shaped things that can carry the oxygen for us. All right, on a side note, I'm going to tell you the shape of this blood cell ties into this statement right here. Okay, the types and quantities of subcellular components might vary between cell types. So our red blood cells actually lack a nucleus. In their development to maturity, the nucleus is actually, they start off with the nucleus, but it gets squeezed and popped out of them. And that's what creates that little disc right there, that little shape. So these guys actually lack a nucleus. All right? Our muscle cells. Any idea for these cells that are doing a lot of work, what type of organelle you might find a lot of?
Ribosomes.
Okay, why ribosomes?
Because they make proteins.
Okay, they might have a lot of proteins. What else?
I don't know.
They're doing a lot of work. They exert a lot of energy. What makes energy for your cells? You guys gotta go look at your organelles. Go back and study. What is it?
Carbohydrates?
Oh, well, so what organelle takes the carbohydrates and breaks it down to get energy out of it?
Well, look at this one.
All right. What organelle makes ATP?
The
site of cellular respiration. You guys gotta go learn your
the
organelle.
mitochondria.
Mitochondria, right? So
The one that looks like a potato chip. I mean, Cheeto.
yeah, the powerhouse of the cell. But we want to know that it makes ATP because it does cellular respiration. Okay. So you find a lot of mitochondria in muscle cells because they need a lot of energy to do what they do. Okay. All right. So keeping that in mind. Next, so hopefully you guys had a couple things jotted down in there. Last question, okay? Again, an AP College Board question. It would be a short answer or short response in either on the website or in their test in the spring. But we're going to compare different types of cells based on the differences of organelles, okay? So the following data were collected by observing subcellular structures of 3 different eukaryotic cells. Relative amounts of organelles in 3 different types of cells, okay? What we're looking for here is based on the types of organelles that we find in these different types of cells, is identify the primary function. I'm not looking necessarily for a cell type, like you don't have to tell me nerve cell or whatever, but I want to know a function of these cells and explain how the data supports that, or why do you think that? Right? So identify and then explain why you think that. So for the first one, if I have a cell that has small amounts of smooth ER, small amounts of rough ER, but a large number of mitochondria, ding, 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 we just talked about that, right? Cilia present, what's cilia? That's flagella, close. The cilia are like the finger-like projections, right? And then small amounts of Golgi body, what might be the function of this cell? To make energy, and that energy might be used for movement, locomotion, okay? So good, this one might have to do with movement.
So for the function.
Or you could even say muscle contraction.
For function, put muscle contraction or to create energy.
would say the function would be movement. And what you're going to do is in the explanation talk about the fact that the large amounts of mitochondria produce the energy that's needed for that movement. Okay? All right, let's look at the next one. For cell type Y, we have large amounts of smooth ER, large amount of the rough ER, a moderate amount of mitochondria, don't really have any cilia, but we've got large amounts of Golgi body. Guesses?
To modify and create protein.
Perfect. Protein production, right? They're there to make some kind of protein for the body or for the organ. So protein production. And your explanation might have to do with the fact that the rough ER and the Golgi body sort of work together to make and modify proteins. Again, this is kind of showing you how you kind of have to know how things are working together. Kind of have to know that, okay, if I'm making a large amount of protein, I'm going to need the Golgi body to do its job with it, you know what I mean, and package it and get it ready for export. But on a side note, smooth ER, what else might be being made here? What's the job of the smooth ER? It doesn't really make proteins, it makes something else. You guys remember what smooth ER makes? You know how I always remembered smooth ER? Because it makes lipids. And lipids are fats. And I think of fats as being oily and smooth.
Oh.
Okay?
I know what you're trying to say now.
So that's just my way of making that connection. Oh, the smooth ER makes the smooth stuff, the fat stuff, right? The lipids. So it makes lipids. So we got lipid production going on here too. But just as a refresher, I'm just trying to reiterate our functions of the organelles. What else does smooth ER do besides make lipids? It does one other thing. No? Nobody remembers? Detoxifies. Gets rid of toxins. You guys remember that at all? Detoxifies? So I'm going to follow that up with, can you think of any other type of cell in the body that might have large amounts of smooth ER because of this function of it? What do you think?
Your kidneys and your liver, because your liver filters blood.
Yeah, so it's more of the liver. If you look at the liver cells, tons of smooth ER in your liver cells because its job is to detoxify. So it's got a lot of that. It's got a lot of that in there. Okay, good. All right, Z is kind of a tricky one. What do you think?
Wouldn't it be nothing?
Okay. So it doesn't really do much, right? Because there's not much. There's no organelles there to do something. So what might be the purpose of a cell that doesn't really have much to do? It's just kind of sitting there.
To fill space, like, and make skin appear and stuff like that. Like, it doesn't do anything, it's just for a color.
Okay, storage, right? It might be storage based on what you're saying right there. Like you said, for a color, we do have, like, plant cells have certain cells that are just there to store pigments. So they don't really have, which gives them color, right? So they don't really have a whole lot of other stuff going on in there because really they're just a holding cell, right? So it might be for storage. There's another cell type that we have in our bodies called adipose tissue. And adipose tissue is fat cells. And they don't do much except store fat for you. Some people have more adipose cells than others. Okay? And so their job is to store fat. Again, wouldn't have much of the other organelles going on there. There's other possibilities here though. So, Anita, what did you say originally?
It was nothing because it's absent.
Okay, so what might be going on here if the cell's not doing anything?
They might not
It's
be
dead.
needed.
It could be
Oh.
dead.
That as a copy.
It could be a dead cell.
I was gonna say it could have been also a prokaryotic cell because
Okay.
they don't really have anything. that
They
has
don't have
nucleus.
specialized organelles, so that would have been a logical response as well. I would have taken that. They do have to do cellular respiration, but they don't necessarily do it within the mitochondria. That process is just kind of taking place inside of the cells.
Look at you.
All right? Perfect. So you guys have been filling in the next slide, which is awesome. Once you're done with that, you can submit it to me. Hopefully you get the idea. You got to know how things are working together inside of the cell, and that different cell types have different structures and different subcellular components based on what they do. Okay?
Do your English one?
Not just the beginning.
And that's it.
I did the first two.
Miss, for the mighty pig, can we speak